The topic of cellular adaptations, injuries, and the genetic control of cell function addresses critical aspects of cell biology that are foundational for understanding health and disease. Cellular adaptation is a process by which cells adjust to stress or threats to their survival. This can occur through various mechanisms, including changes in cell size, atrophy and hypertrophy, cell number, hyperplasia, dysplasia, and anaplasia, and cell type, metaplasia. These adaptive processes enable cells to survive in altered conditions, though they may also be precursors to pathological states. Atrophy is characterized by a decrease in cell size, often due to factors such as immobilization, loss of nerve function, diminished blood supply, inadequate nutrition, loss of endocrine stimulation, and aging. Despite the reduction in cell size, the cells are not dead and can still function, albeit at a diminished capacity. Conversely, hypertrophy involves an increase in cell size in response to increased work demands. This is evident in conditions such as the enlargement of cardiac muscles in response to hypertension or aortic valve stenosis. Hyperplasia, another adaptive response, refers to an increase in the number of cells in an organ or tissue capable of mitotic division. It can be physiological, as seen in breast and uterine enlargement during pregnancy due to estrogen stimulation, or pathological, such as endometrial hyperplasia caused by excessive hormonal stimulation. Metaplasia involves a reversible change in which one adult cell type is replaced by another type better suited to withstand chronic irritation and inflammation, exemplified by the adaptive epithelial cells in the trachea of cigarette smokers. Dysplasia is a more concerning form of adaptation characterized by deranged cell growth that results in cells of varying size, shape, and appearance. It is often a precursor to cancer. Cell injury can result from various physical agents, including mechanical forces, extreme temperatures, and radiation. Physical injuries such as contusions, lacerations, and fractures can result in direct tissue damage, while temperature extremes can cause damage by altering blood flow and cellular metabolism. Radiation, particularly prolonged exposure to X-rays and other ionizing radiations, leads to nuclear alterations and damage to DNA and cell membranes. Chemical injuries result from exposure to substances such as lead, carbon monoxide, ethanol, and mercury, each of which can interfere with cellular functions in specific ways. Nutritional imbalances, whether due to deficiencies or excesses, also significantly impact cell integrity and function. Vitamins and minerals are essential for normal cellular operations, and imbalances can lead to diseases such as kwashiorkor and atherosclerosis. The mechanisms of cell injury are complex and multifactorial, often involving free radical injury, hypoxic cell injury, and impaired calcium homeostasis.